Shall we wait 5 minutes or what time were we supposed to start? 9 for the announcement. We have time.
Okay, good morning, everyone. I hope you had a nice evening yesterday. Well, you survived. Okay, I have very few announcements to make. Three actually. The first one is about the famous badges. Please, can you before you leave, can you hand them back to the organizers somewhere in the hall, or okay, because we keep them and we use them. We will reuse them for the next Euring meeting. Second thing, for those of you who have to leave before this afternoon, we've arranged lunch boxes for lunch, food again. You can grab them at the usual place. The soonest will be 11:45. Okay. So 11:45, you can go down the campus and grab a lunchbox for you and leave if you have to leave, okay? We couldn't let you leave without more food. The last one, the posters. For those of you who had a poster in the hall, don't forget to take it back, okay? And thank you very much. And I will leave the mic to Thomas. Thank you very much. Enjoy the last day of the conference.
What a tremendous pleasure and honor to introduce Dr. Todd Arnold from the University of Minnesota. A couple days ago, I asked Todd if there was anything he wanted me to say during his introduction, and he said, just please don't say anything embarrassing. So I compiled comments from dozens of Todd's undergraduate students over the years, and were unedited. Todd's students said that he was kind, caring, attentive, passionate, incredibly interesting, always willing to listen and help, and thought-provoking. I couldn't agree more, and it's such a pleasure to have Todd as a mentor and a friend. I'm so excited to hear his talk: density dependence, individual heterogeneity, and demographic compensation, new approaches to age-old questions.
Thank you and good morning. And even though I wasn't at the last Euring meeting, this has been my favorite meeting so far. Euring meetings always are, and I'm so honored to be asked to give this presentation. Back in January, when I was asked to give this presentation, I had very grand, eloquent ideas about what I would present about. By the end of last week, my perceptions had changed a little bit, perhaps. Yes. The motivation for some of what I'm going to talk about, the funding that has supported me over decades, has been in what I call the Mallard Industrial Complex in North America, and my own interest in, say, optimizing, understanding harvest in mallards on any given day might range from intellectually stimulated to bored shitless, and maybe with a very flat prior. And I suspect that a lot of people in the audience, your prior might be piled very much in the other direction. You might even be ethically opposed to hunting mallards. But I want to suggest that the ideas I'm going to talk about are very relevant to understanding anthropogenic mortality in any population, if we can measure population dynamics, and if we have a very good measure of some source of mortality that we're interested in, these ideas apply. They also apply to, they apply to predator-prey situations if we want to understand how multiple predators might impact a prey population. So there's sort of three topics I want to cover today. and I want to look at how can we estimate the effect of a mortality factor such as harvest on population dynamics. If we find evidence of compensation, I think the onus is on us to try and demonstrate how does that compensation occur. And there are a couple of mechanisms that have been identified. One is density dependence. The other is individual heterogeneity. And so I want to spend a bit of time on each of those and ask how can we measure these things. So let's start with the classic models, and this is going back to maybe Anderson and Burnham, 1975, additive versus compensatory mortality. And I want to point out that there's only one key parameter to understanding how these models work, and it's S0. What is survival in the absence of hunting? A little bit of a paradox there. We're estimating survival from dead recoveries of banded birds that we can't even estimate survival in the absence of harvest, but let's forget about that for now. S0 is the intercept when there's no harvest. I think we'll all agree that if we harvest them all, they'll all be dead, so that's an easy point to put on the map too. And S0, negative S0, is also the slope of that relationship. S0 is also the proportion of survival that's additive, because 1 minus S0 would have died anyway. S0 also helps us identify the inflection point for compensatory mortality, where the slope changes from 0 to 1. So to me, the key is we need models that can estimate what is survival in the absence of hunting to distinguish between these two models. So if we've got survival probability of surviving harvest mortality, we can think of a mortality probability as 1 minus survival. Better maybe to convert it to an instantaneous mortality rate. Similarly, survival in the absence of hunting, natural mortality, I called it S0 in the previous slide. We can have a mortality probability as 1 minus that, or instantaneous. If we want to get total survival, that's easy. That's the product of the two survival probabilities. If we want to get total mortality, do not do this, please. And Anderson and Burnham did this. They acknowledged it was wrong. People have continued to do this through 2023. I will not name names, but you cannot die twice. And so if you want to do that, the approach you should take is something like this, kind of how we estimate P star in occupancy modeling, if you're familiar with that idea. But better yet, work with instantaneous mortality rates where they truly are additive. There's nothing new I'm pointing out today that wasn't pointed out by Jean-Dominique in this wonderful paper. All that's new, we maybe have some better techniques for measuring some of these things. In this paper, you pointed out, I think very correctly, that we cannot have such strong density dependence, such instantaneous density dependence to allow total compensation. So let's take that one off the table. But importantly, even weak compensation can have a really important influence on population dynamics. So to me, the challenge is not to distinguish between additive and compensatory, but to distinguish between additive and partially compensatory. And so, to my mind, models that satisfy the assumption of staying inside of this blue triangle, broken stick models. They're not really maybe biologically relevant, but they can keep estimates of survival within this parameter space. And you might be aware that we could have super additive mortality, we could have super compensatory mortality. For the most part, I think that has interested theoretical ecologists in the real realm of things, at least that I work in. I think we're trying to identify stuff within this realm. So I wanted to evaluate a bunch of different models and see how would they perform with simulated data. And one approach we could take is just do a classic Brownie model, and then post hoc in a second step, work with the point estimates and fit a regression. And we can do it fancy. We can recognize variation in X and variation in Y. We can even fit broken stick models to those point estimates. We can have the pure additive model. We can have the additive model with annual random variation in S0 to approximate what might be closer to reality. We can have a model that models a process correlation between annual survival and harvest mortality, kind of work that David Otis and Gary White developed in earlier Euring meetings and many other people, including myself, have used. We can have the compensatory model, compensatory plus random variation. We can use hazard rates. And here I've converted them back to survival, so they're sort of on the same scale. Can fit a broken stick model or a broken stick with temporal variation in S0. Notice if you have temporal variation in S0, the challenge of identifying the functional relationship is exacerbated by the noise. And here's an example of fitting those 9 models to simulated data. And I guess one thing that jumps out at me, if you look at the leftmost and rightmost column, is that anytime we add random effects to a model, it allows a model that's functionally wrong to fit the data pretty well. I don't know, I think of this as being like in dim lighting, we're all supermodels, and maybe random effects are a little bit like that. But you can really miss the functional relationship. So how do these models do at identifying S0? And what I would say is that the pure additive models always overestimate it, and they miss it badly. Pure compensatory models underestimate, and they miss it badly. And the other approaches seem to do a reasonable job. All of the modeling approaches that estimate temporal variation in natural survival seem to overestimate it. And that's a concern, even the ones that do best. And I was really surprised that the post hoc approach of analyzing brownie estimates in a second stage actually seems to be best. Most of the models do quite well at estimating the individual survival rates, except for additive and compensatory models. If you're familiar at all with, I shouldn't say this, but if you're familiar at all with adaptive harvest management for mallards in North America, it's something that's been touted as a paradigm of how we should do things. It's really choosing between the worst and the penultimate worst of the 2 models that are available. I don't think that's progress necessarily. All of the modeling approaches seem to do a pretty good job of estimating a breakpoint of when harvest becomes more additive, except when we get very close to the origin. And what I would point out from my simulations is that when harvest was less than 5%, there would be 6 or fewer data points to the left of the breakpoint. And I didn't consider models that maybe ask, first, is there a breakpoint? And if there is, where might it be? The thing I'm most interested in estimating is what's the slope of that relationship initially. And here we didn't do so well. And we're overestimating the proportion additive at when it's low. We're underestimating when it's high. I think part of this might be the prior structure I used, and that I allowed it to vary from 0 to 1. So it's a bit of a concrete wall on both sides, and maybe I need to allow for super additivity and super compensation with my priors. But to me, this is, from a harvest management perspective, this is the parameter we want to estimate the most. And there's some potential there, but it needs more work, I would say. So moving on from this, if we have evidence of compensation, I think the onus is on us as quantitative ecologists to demonstrate a mechanism by which it could occur. If it's additive, that's just pretty straightforward. You harvested them, they're dead, they're not there anymore. And I think the tools for estimating this have gotten much better by having access to integrated population models. Another plug I would make is that we should use instantaneous mortality rates. I try to reread this paper every year. It deserves a whole lot more love than it's gotten. Please love it, cite it to death. And I don't know, after Tuesday morning's talk, Mateo's talk, I thought, oh, wow, there's some monsters here in terms of estimating density dependence. And I stopped adding updates from all the rest of your talks. But what's really wonderful about coming to a Euring meeting is that you realize how much stuff you have to change because of what you've learned in the last 2 or 3 days. This is some work that Thomas led and I'm part of that tries to get at some of the density dependent interactions. When we work in waterfowl, we have to hide what we're working on in the title so that we can get it published somewhere cool like Journal of Animal Ecology, but this is actually on mallards. And I'm going to label some of these things so they're easy to see what they are. But the model structure that Thomas used looked for evidence of density dependence in natural mortality and evidence of density dependence in fecundity, and found evidence of density dependence in fecundity and adult female survival. And that's actually the mortality probability or mortality rate rather than survival. So tipping upward is evidence of negative density dependence. So in 2 out of 3 cases, there was evidence for density dependence. And I think the onus is on us if we're going to suggest something is compensatory to try to find a mechanism by which it could occur. And I'll go back. I love this approach. The one thing I do a little bit differently in hindsight is use log abundance on the x-axis. If we can partition mortality into seasonal components, I think it makes our job easier to try to find evidence of density dependence. And so I was excited by talks that took us this direction, Clark Rushings and others. And if we mark individuals at different time periods, we can estimate seasonal survival, even from dead recovery data. And so using post-season bandings, post-hunting season bandings, and pre-hunting season bandings in black ducks, we can partition annual survival into seasonal components. And sort of looks like this. And here's a model that came out of it. When I first fit this model, just sort of, for fun or something, I fit it as instantaneous mortality and instantaneous fecundity, just for something different. But I actually think that's the best way to consider this. The balance equations look like this, and we subtract instantaneous mortality to get the new population size. On the log scale, we add instantaneous fecundity, we get the new population size post-breeding. And what I want to call your attention to is the instantaneous mortality rate produces a difference in population size on the log scale. And that's our metric of what we have to look for in terms of compensation, if compensation is going to happen in terms of density dependence. So it would have to be extremely strong. We'd need a slope of, and this is instantaneous mortality, so it'd be -1 if we were looking at survival. In the simulation Mateo did of strong density dependence, he used -0.3, and moderate was -0.1. A relationship of 1 is literally impossible. And I agree with what Jean-Dominique said back in his 2005 paper, that it's literally impossible to imagine density dependence is strong. But we can get components of it from different parts of natural mortality or from fecundity. And let's not forget about fecundity. I suppose we could even get it from immigration, but if you're having, I don't know, a sustainable harvest of brown bears in Norway by stealing Sweden's brown bears, that's really not sustainable in the bigger picture. So let's forget about that. In the black duck data, the relationship we need to see with natural mortality is in red. The relationship we see is not providing us with that strong a signal. I had questions when I made this slide, and I actually made this slide while I was still here, but before your talk, Marlene. And what is the x-axis? Should that be adult female? Should it be the entire population, and now I know it should be yes, yes, plus a few more things if possible. But we don't see evidence of the density dependence it would take to produce compensation in black ducks. Okay, what about the role of individual heterogeneity as a final thing to look at? I like to use finite mixture models. I call them pleasure models. They're fast. You can fit them very quickly. You can fit them in an exploratory context and look for evidence of heterogeneity. And if we don't use it on the classic dipper data, we get an estimate of survival without heterogeneity that looks like this. If we consider heterogeneity, we find a small component with very low survival. The classic San Luis male mallard data, no heterogeneity, estimate of adult male survival there, add heterogeneity, and we get two components. Again, one with a very small component of survival. Doomed surplus. I like to think of these as the Walking Dead. And in the models that follow, I'm just going to push them farther to the left and give them zero survival and ask what the component is. That makes modeling easy if you die and go away. But I'd also point out that what's the sustainable harvest rate on zombies? You can harvest them all is the answer to that. So what is this doomed surplus? And is there evidence of a doomed surplus in populations of harvested species? So I was very excited by Emily's talk, and I can't remember what day, where she looked at a variety of European species. And I did something similar with a variety of North American species, dead recovery data, and asked, is there evidence of heterogeneity in survival from band recovery data? And for 8 out of 12 species, there was strong and compelling evidence of heterogeneity in survival, typically involving a component with expected survival near zero. So what is the implication of that? I explored the implications of that by imagining that each of these two groups had a similar vulnerability to harvest, and so that harvest would be spread across both groups. And for 4 of the examples, here's what it looks like when we consider heterogeneity and when we ignore it. When we ignore it, our estimate of S0 is survival in the absence of hunting, and that's also the slope of the relationship between harvest mortality and annual survival. So, minus 9.06 for Canada geese, that's very additive level of harvest. When we consider heterogeneity and assume equal vulnerability, the leftmost number is our estimate of S0, survival in the absence of hunting, and the rightmost number is the slope. And notice that they're weaker, they're substantially weaker. And so I would argue that in these species, heterogeneity provides a mechanism for compensation. I'd also point out that compensation could be increased if that most vulnerable component is also more vulnerable to harvest. The Brownie recovery rate or the Sieber recovery rate, though, is a Bernoulli. We see it one time. I'm going to argue that we can't estimate heterogeneity in that parameter. I'd love somebody here to prove me wrong, but I've beat my head against the wall enough times that I'm pretty convinced we can't. But even if we can't, it provides a mechanism for compensation. If those individuals are even more vulnerable to harvest, we get more compensation. If they're less vulnerable to harvest, we get less opportunity for harvest. I mean, the worst case scenario would be that those vulnerable individuals all die before hunting season begins. We don't observe them, and we're harvesting the fittest part of the population. So I'm interested in all 3 of these things. I think they're important issues to resolve. I think there are a lot of uncertainties about all of these, and I've listed some here, and I don't want to leave this slide up too long, but actually I do. I want you to add to the list. There are things we need to be doing with this. And I'll close by saying that I got to go back and teach, unfortunately, but then I'm going on sabbatical. And I want to, in the year I have off, I want to address some of these issues. And I want help addressing these issues. So if you're interested and you want to get involved in any or all of these, let me know, and I'll stop there. Sarah has her hand up, way in back. Did you get some sleep last night, Sarah?
Thank you for a great talk. I was just wondering if sometimes heterogeneity is not something that is changing through the life of individuals. And so you say that maybe this certain class of individuals would contribute more to the population and give more potential for compensation. So I was wondering if you consider age how age could play a role, because I imagine like individuals that have good potential to produce many offspring, for example, but if they get hunted once they finish their contribution to the next generation, like trophy hunting or something, when you hunt the very big ones, but they've already contributed. So yeah, so I don't know exactly what my question is, but it's something around the effect of age and heterogeneity, how this could play a role here.
But it's an important component of how heterogeneity models work. And if it's an individual random effect, you're presumed to have that benefit or detriment for the rest of your life. And I don't think that's very realistic. And notice what I did is I slid survival down to 0, and I just got rid of those individuals. So I didn't have to worry about what happened to them next year. I just pretended you all died. And that's probably not realistic. The examples you give about trophy hunting, I think there's definitely potential there to remove not the most vulnerable, but maybe the most important individuals. And I think of examples like in wolf packs, if you lose the alpha individuals, the entire pack can disintegrate. Take out a male brown bear and a new one comes in and triggers infanticide events. That happens in lions. So I think we can imagine scenarios that could be super additive as well. And the heterogeneity part, to be honest, is where I most want the help, but I want the help everywhere.
Thanks a lot for the presentation. When you look for heterogeneity, do you typically find this pattern of some a small part with very low survival, or do you have some species for which you find a few portions with very high survival, for example? Is this pattern constant, or how does it vary?
That's a good question too. The typical pattern that I've seen is for a small component to have very low survival. There's no reason why you have to stop at 2 mixtures. You could potentially have 3 mixtures, and sometimes the 3 mixtures suggest that there's a small component of the population that are like Greek gods and immortal and they never die. And in a dead recovery situation, we never see those again. So I think that's an artifact, but who knows? I'll leave a little room for magic in my talk too. What I'd maybe like to do with heterogeneity is combine the 2 approaches, and can we identify a small component maybe of super vulnerable, but then consider individual random effects for that more typical survival? Because I think that approach would make sense. I have not had luck fitting that sort of model. So again, that's a place where I could use some help.
I really liked your talk. I was wondering about the individual heterogeneity and the small proportion of individuals that had really low survivals. And is there any possibility that could be like tag loss or handling mortality?
It wouldn't be tag loss because it occurs in the very first year, the very first hunting season following ringing, and so it's unlikely there. Tag loss maybe could explain why some individuals seem to be Greek gods and they never die. Well, no, they lose their bands and we don't follow them anymore. In North America, we need better bans on long-lived species. I've heard ringers complain that the US has the worst made bans of any country in the world, and I don't know that that's true, but we could talk about that afterwards. So that was one component, and I think I missed the other one.
Handling mortality.
Yeah, handling mortality. I think that's always an issue, and concerns about capture myopathy and whatnot. There are no additional markers on these birds. I think anytime we put something larger like a radio transmitter or a GPS transmitter, we really have to be concerned about that. Although in those cases, we also typically find them quite quickly, and we know that, oh yeah, this is not a good package to put on this individual.
Thanks for the talk. I find it quite striking that this pattern of this small group of doomed individuals seems to appear so consistently. So I was just wondering if I assume somebody checked if the finite mixture models can produce this pattern as an artifact.
I've done a lot of simulations comparing individual random effects with finite mixtures. And if you tried both approaches, they do a pretty good job of identifying the correct distribution. Olivia, you've done a lot of work with these models as well. Yeah, you have. And if nothing else, I'd say they're fast. Use them to see, do I have evidence of heterogeneity? And if you want to go down the individual random effects road, you can do that. Be patient if you do that.
Well, maybe a brief comment on finite mixtures. In fact, finite mixtures with two components do extremely well to represent any type of heterogeneity. It's used well beyond the capture-recapture stuff. It's basically to estimate the average, the mean, and the variance of a phenomenon with a two finite mixture is as good as a continuous, so the individual random effect. And usually the three-level mixtures brings very little and has some side effects, as you say. So there is a lot of literature on this.
Yeah, and in the early days of doing simulation modeling, there are a lot of examples of using finite mixtures as a way of understanding, what's the concern about violating assumptions about the III assumption of identity of rates and independence of fates among individuals.
Yeah, do you know at which age these doomed to die individuals die? So I was wondering whether it's like, it could be a critical age where mortality is increased, and maybe this could be density dependent, and then that's how this heterogeneity can contribute to compensation.
It's difficult to fit this model to multiple age classes, and the difficulty is in juvenile survival only happens once. They're only recovered once. Adding an additional parameter on that, it's not identifiable. And so to do the mixture model, you have to ignore looking at age effects, and age effects exist. To do transient models, you have to pretend there are no age effects in survival. And so anytime you put an additional term on that first diagonal, you lose the ability to estimate something else. And so I applied these to adults only, and where I would really expect to see the doomed surplus is in the new cohort of recruits, a lot of dumb, young, teenage, you're going to fly south and you're going to see some duck decoys and think this looks like a good place to land, and it won't be. I feel like this is my PhD oral exam up here.
I'll throw you a softball. Do you think at all about, in the systems that you think about the role of environmental heterogeneity and how that might mask any density dependence, and whether adding a covariate or adding an effect that's related to other conditions might suddenly, I mean, I know in a lot of the systems I've worked, you have to deal with some other environmental factor that varies widely before you can start to see the density effects.
Right. Thank you for that question, because I think that's a great question. And to estimate density dependence from time series of counts, we know that it takes a lot of data. And so if it takes 30 years to have a reliable chance of estimating density dependence, if it occurs, what's the probability that carrying capacity has remained static for 30 years? I don't know, but I'd argue it's nil. One of the things I liked about Thomas's model with mallards is he included pond numbers on the prairies. And I think that's a proxy for carrying capacity going up and down with habitat conditions. And for animals in temporally varying environments, I think that's a key component to think about. Density dependence, I mean, what is it? It's competition for something, and we should maybe try to identify what that something is and try to measure it on an annual basis and use that as a covariate. Well, thank you very much. This was a pleasure.
Yes, so it's time for a break. Thanks to the government and
Well, you know, it's nice to have that information. It would be very better if you have information on the baseline. It's a little bit that's what we found first. A few animals smaller. Yeah, that's hard. Yeah, that's all right. So I the main value of that model. I thought you could combine it with some other information. Forget to bring my maybe send me an email sometime. Let me know how your work goes. Okay. Yeah, that's an important question. You see your PhD.
Have another computer to check all the questions. It will be said.
It might be looking to find someone to start in November. But it's flexible. Contact you. Are you on WhatsApp?
Toi qui présente?
Oh, maybe I'm gonna scan yeah, I'm using it as a no, I'm on there. Yes. Yeah, and that one's to try to post so it's way too much here and body weight. So we're trying to model the dynamic. Yeah. Okay. Maybe shoot me a message so I have your contact and email. She does amazing work, yeah. I'm always impressed by it. Yeah, that's very, yeah, that'd be very okay. Go. Nice meeting you. Yeah.
What does he say?
So it says
It takes a few minutes to load. So maybe can we pull it up? Yes. Let's wait for the beginning. Silence. Yeah, 12 minutes. Yeah. About the setup. I have no idea about the audio setup, so I think it is okay. But it says, "Are you getting any sound?" Yeah, so nothing, I don't think so.
Alors les gens qui sont en Zoom, ils devraient être. Ah oui, mais parce que t'as mis le son du PC, ouais.
Port design of marine ecology.
Moi, ça m'a pas fait. Tu sais, le truc, tu te prends le niveau des jeux, en fait, c'est juste comme tout est coché.
Ouais,
voilà, c'est ça.
These and others that are probably fetch our package EMON. My name's Stephen Gregory, and I'm back.
It looks like it starts from the beginning, but we'll see. 12 minutes is a bit long.
Not too bad.
Check, check.
The microphone is on the.
Hello?
Okay. So welcome back, everybody. Good morning. Welcome to our first session on management and conservation on the last day of the conference. Our first speaker is Stephen Gregory, and his talk is Power Analysis for Real-World Sampling Strategies.
My name's Stephen Gregory, and I'm presenting today on behalf of myself and John Barry, work that we've been doing to update our package EMON to do power analyses for real-world sampling strategies. Now, I had a quick look over the booklet of abstracts and saw that there are plenty of very interesting capture mark recapture studies, among others, that are probably fairly heavy on maths and equations. And so some of you this Friday morning, might be pleased to hear that this one is going to be very, very light and probably quite short. I don't think there's a single equation in here. So let's have a look. Long time ago, back in 2014, this paper was published by John Barry et al., introducing a new R package called EMON. What does EMON stand for? Environmental Monitoring. It's an R package that was designed to support the design of marine ecological and environmental studies, surveys, and monitoring programs. It was first released in 2014 to RForge, and in 2017 it made it to CRAN. And since then, it's been slowly accumulating downloads, and now in 2023 there was something in the region of 20,000 downloads or something like that. So it certainly seems as though people are looking at it. But a lot has changed since 2014 and even since 2017, including a large amount of interest in generating offshore wind energy, amongst other things. The list could go on, but I just chose that particular issue because power analyses are required when doing offshore development planning offshore wind farms. You'll need to do some sort of environmental impact assessment. But also R has changed a lot. Back in 2017, I think we were on version 3.3.3, and now we're over version 4.3. So it's gone up a whole large version since then. So it was about time that EMON got with the times. So we've introduced some new functions to EMON. Now you could call it EMON 2.0, although it's just I call it EMON. The first one is power analysis of designs with replicates. There are other functions, but I'm not going to mention any of them here today, for there isn't time. We've also introduced some improvements. We've optimized a lot of the existing code, and we've optimized the new code using vectorization and parallelization. We've extended plotting facilities to include the possibility of using ggplot. And also we're trying to work on standardized reporting of power analyses as well. And I'll talk a little bit about that towards the end. So what is power? We're trying to answer a question like how many samples are needed to identify a change in Y in either space or time. Often a power analysis requires us to make some guesstimates or use existing studies to come up with best guesses of the following information. We need information on the expected change in Y between the first and second groups of samples, the variance in Y in the first and second groups of samples, and the numbers of samples that we'll have in both groups, the first and second groups. Now, EMON does power analysis via simulation, a bit like this. We have this user-defined information, the information I just went through, and probably some other information as well. That's used to parameterize a data set called T0, for example, and T1, which will be the surveys in the first time step or in the first location, and then in the second time step or the second location. Those data are combined. And basically, we fit two models to them, a treatment GLMM, a null GLMM, and we compare the fits of those two GLMMs, and we do this over n times. And at the end of that, we look to see how frequently the likelihood ratio test prefers the treatment GLMM compared to the null GLMM, and that is a measure of our power. So what is a design with replicas? What am I talking about? Well, in this little figure here, we can see a bunch of X's where samples are taken, and you can see that they're assumed that they're sort of randomly, they're treated as random samples. Each of the X's has a subscript and they go between 1 and 15. But these have been taken in groups, and now the subscript of the X's are in groups 1 to 5, and in each of those groups there's 3 samples. So we've got X11, X12, X13, all the way through to X51, X52, and X53. Increasingly seeing these kinds of sampling strategies, probably to save money, because once you're parked up in a boat, it's probably worth taking a few samples rather than just taking the one. But if we assume that those samples are independent, then we risk running into issues like pseudo-replication, which may bias our estimates of power. So if you take a simulated data set which has this grouping, or these replicates are designed with replicates, and you fit it with GLM and GLMM using EMON, then you, for different changes on the x-axis, a minus 20% change through to a 20% positive change in the samples between time 1 and time 0 and time 1, then what we find is that the GLMM will always give us better statistical power. And that's because it's taking some of that variation within groups and dealing with it as a random effect, taking it away so that we're clearing away some of the variation, so we can look at the variation that's due to differences in the treatment or the time effect. So when we're doing these designs with replicas, we're taking into account this grouping of the sample. So we're looking at a model something like this, where Y is a function of a treatment, that is whether it was taken before or after in time 0 or time 1, and whether it's part of whether each observation is part of a group. So for example, that would be, we've got the X11s, X12, X13 would be one grouping, and here we'd have a group of X1. Then we'd have group X2, group X3, right the way through to group X5. These are generalized linear mixed models. I'm sure you're all fully aware of this anyway, so I'm probably wasting my breath here. So understand then that they're fairly complex models, fairly slow to fit. So there was a need to optimize a little bit to make sure these sorts of power analyses wouldn't be obscenely intensive, waste too much time, to parallelize the problem. As I'm sure you all know, your computer is made up of a bunch of separate processing cores, and so you can take your data set, split it up into in a bunch of chunks of data, one for each of your computer cores, and then you can reduce the amount of time your computer takes to do those computations. And really the gain in time, the efficiency is quite dramatic over the first few cores at least. Emon does that. And because Emon does that, because it's fairly efficient and it can fit these GLM power analyses fairly efficiently, we can start to look at a bunch of different possibilities. So I'm saying here that it's got flexible support for replicate designs. We can look to see how, if we were to change the numbers of sample locations, what would that do for our power? And this plot here is just showing on the x-axis if we had 30 samples in time 0 and 30 samples in time 1, with the other information fixed, 10 replicates, for example, and a change of minus 15% on a mean of 20, then we get a statistical power of about 22%. If we were to reduce the number of locations to 20 in time t and 20 in time 1, then we drop in statistical power down to below 15%. And so you can look at different combinations of numbers of locations and see how that could affect your statistical power analysis. So change the numbers of replicate samples per location, just as we did a moment ago with locations. And again, you can look to see what the implications of that is for your statistical design or your sampling design. And you can do lots and lots more things as well, because it's fairly efficient and because it does this fairly quickly. So standardized reporting, we see a number of environmental impact assessments for different offshore developments, marine developments, but also extractions and all sorts of other different things. And there doesn't seem to be a great amount of standardization, which makes it a little bit tricky to assess the quality of what's been done. So we're in the process of designing a standardized reporting scheme using R Studio, possibly using Shiny, possibly using Quarto, coming up with different templates and things to help people come up with standardized reporting. So the idea will be that they either upload data and use Emon, or just use Emon locally, and then there'll be some sort of perhaps standalone process using R and R Shiny, or even WebR, this new browser implementation of R. Perhaps we'll have something hosted either at CFAS or somewhere else on a UK government website where people can upload and use Emon. They can write the report text and submit that also to the same service, and that could be written either in R or in Quarto, which can also include interactive elements if needed. Download that report and then send it off to review. And we're keen that people do this, because then we can ensure that the code that is used to analyze the data is all presented, and the data that are analyzed are all presented together along with the report, so that we can make a thorough and easy assessment of what's been done. So that's a quick whistle-stop tour through a few updates to Emon R package. There's more in the pipeline, and we're currently preparing a manuscript for submission to the PCI journal, so please look out for that in the not too distant future. Thanks very much for listening.
I think we have time for one brief question.
Yeah, thank you for presenting Imon. It's nice to see that life gets easier. Does the package report precision beside power? Because I think it's way more useful to, or I normally decide for a sample size based on precision rather than power.
Can you hear me okay? Thank you for the question. It's a very good point. I think it should. It doesn't currently. Like you say, I think it's actually potentially more important to understand uncertainty in that power than just the power itself. So that's something that we'll look into doing.
Okay, well, thank you so much, Stephen.
Thank you.
Our next speaker is Dr. Mikkel Schau, Lessons to be Learned by Comparing Stock Assessment Methods with Integrated Population Models.
Okay, thank you, Thomas. This work that I present today is a collaboration with my good old friend Mark Carey and people from fisheries. So integrated analysis have become the gold standard in fisheries and wildlife population ecology. In fisheries, we talk about integrated stock assessment models, whereas in wildlife biology, we talk about integrated population models. The development of these models or the types of models occurred largely independent from each other. The fisheries stock assessment models have been developed about 20 years before the integrated population models. And these models are very similar. They have a lot of share a lot of things, but there are also some differences. And here, what I want to do is to show you very briefly and also superficially a stock assessment model, which then allows to compare it with an integrated population model. I assume that you know integrated population models because there have been several talks about them, and so I will not show an integrated population model. And then we will see whether there are some things that can be learned. So the case study I will show you is about this fish species, snapper. It occurs in the Pacific Ocean. The data sets that have been collected off the west coast of New Zealand, and there are 3 different data sets that have been collected. The first one is the catch in biomass. So this is the commercial data, so from the commercial fisheries. These data have been collected over a long time. Then there is a catch per unit effort abundance index. This also originates from fisheries, from catch. But this here, the effort is more standardized. So we have a more reliable index of the population development. And then there are age proportion of these captured fish that have been recorded during some years. Now the model, when these stock assessment models, to set up a stock assessment model is very similar to when we do an integrated population model. The core part of this model is a population model which links the population size with the demographic rates. And here they have used an age-structured population model with 20 age classes, and we see here the population size in year t+1 of age class A is a function of the population size the year before of age class A -1 and some survival. And survival here is expressed or is a function of the natural mortality M and of the fishing mortality F. This is also multiplied by the selectivity, which is the age-specific probability that a fish is captured. Because the fisheries, commercial fisheries, they are not interested in small fish, but they only want to get the larger fish. Then the recruitment here, this is usually integrated population model, we would express that per capita, but here it's just like a single recruitment process. So that's the first step. And the next step is to write the likelihood of the different data set, for the different data set that we have. This looks now more complicated, but I will not go into detail. I'll just show, well, there are 3 different data sets. We use, they have used log normal distributions for the 2 removal data set and then the multinomial distribution for this age composition data. And the parameters here, these are demographic rates, population sizes, and some variances. Then the next step is to create a joint likelihood by the product of the single data likelihoods, and from this joint likelihood we can then make inference. Now here in this model there would not be enough information to estimate all the parameters, so therefore some parameters have to be fixed. For example, the natural mortality here is fixed in this model, and then also some variances are fixed. And then can fit the model, we get estimates of population size over time. For example, here we have here an estimate of the fishing mortality, recruitment, and this is the selectivity. So that was a very brief stock assessment model. Now we want to go on and want to compare typical stock assessment models with typical integrated population models, and we start with the context. In integrated population models, they are usually used to understand population dynamics. We want to know whether the population is declining, what are the demographic reasons for that. Sometimes we also want to do a population viability analysis. The goal of stock assessment models is typically, or almost uniquely, of course, to assess the impact of the harvest. We want to know how many fish can be removed from the population without damaging the population too much. The typical taxa on which integrated population models are applied are birds, mammals, sometimes reptiles, and even streamfish. Typically, the populations we are dealing with, they are small. They are also sampled on a relatively small area, which is usually quite well defined. Stock assessment models, they are applied on fish, typically on the ocean, on the sea. Populations are large to very large. The extent of the study area is also very large. It's often poorly defined and may even change over time. Next one is the population model. So with population model, I mean the model linking population sizes with the demographic rates. That's very similar in both approaches here, and this is also, like, I would say, the key part of the model. Usually these are age or stage structured population models. In the case of stock assessment models, we see also length structured population models. Then we get an estimate of abundance. In integrated population models, we often have a relative abundance, and that's fine for the goals that we have. Of course, if you want to make a population viability analysis, we need to have absolute abundance. This is a bit different in stock assessment models because they always need to have absolute abundance, because they are interested in knowing how many fish, or how many fish that can be removed from the population. Other parts of the model, like dense dependence, demographic stochasticity, or environmental stochasticity, are relatively similar. Demographic stochasticity is something typical that it's included in integrated population models, but this is just because usually the populations are relatively small, and that's something that is rarely used in stock assessment models because populations are so large, so there is really no point to include that. Data sets, in integrated population models, we always have one data set, or at least one data set from the population level, which is a relative or absolute count of the population. And then, we have data from the individual level, typically capture-recapture data or productivity data. In stock assessment models, we have the catch, so the removal data from the population, and then usually also need to have age or length composition data. More rarely, we have data from the individual level. Yes. if they are data from the individual well, so for example, capture-recapture data, it's interesting to see that in stock assessment models, they are typically used to estimate abundance, whereas in integrated population models, when we have capture-recapture data, then we use them to estimate survival. Model fitting, mostly based in integrated population models and frequentist in stock assessment models. There may be good reasons for that, but I think mostly this is a bit a tradition. In integrated population models, we usually have no fixed parameters. We also try to use non-informative priors. This is different from stock assessment models where they usually need to fix some parameters or use informative priors. Model weighting is something that is never done. I've never seen in integrated population models, but it's often done in stock assessment models, just to usually to downweight a data set that is large and may contain biased information. So the main difference between integrated population models and stock assessment models appears to be the different objectives, the different goals, and the need of the different parameters to reach these goals. And in a way, the stock assessment models, they need more information because of the need to estimate absolute abundance. And at the same time, it is more difficult to get data from the individual level in the context on which stock assessment models are applied. And therefore, stock assessment models often need more tuning or more tuning, and therefore techniques like weighting or model assessment appears to be more advanced in stock assessment models than in integrated population models. So stock assessment models, they have more strengths for the population level data, I would say, whereas in integrated population models, we typically use quite simple observation models for our count data. And the integrated population models, they have more strengths on the side of the demographic rates. So there could be some benefit if we talk more about, or we could learn from each other. But there are some difficulties in doing that. The first one is the vocabulary, which can be quite different between the two fields. And I think to improve that, we should try to make more transparent presentation, in particular of the population model, but also for the description of the data set, because this is also something difficult sometimes to understand if you look on the other side. So thank you for your attention.
We have time for one question.
Thanks a lot, Michael. I was looking forward to this one. I was wondering, besides like writing better documentation and being more clear and maybe sorting out the vocabulary, feels like talking to each other might help as well. So I was just wondering a little bit, like, given you now have newfound colleagues in fisheries, like, what are your thoughts on how we could promote more exchange and kind of cross-fertilization of ideas and research between the two fields?
Yeah, good question. I mean, I still think like the vocabulary is something important because now we have had some exchange with fisheries people, and it's really also, even if you talk with them, it can be difficult just because of that. And it's not just that we or the integrated population models do not understand them, but it's vice versa. It's also for them, so that they do not always understand what we are doing. And there are some specific terms also that for them it's very clear, for us not, and vice versa. But I do not have really a recipe of, I will hope maybe with this work that can stimulate a little bit more exchange, but I don't know.
Well, I think we don't have time. Thank you so much for your talk.
Thank you.
Our next speaker is Benjamin de Betancourt, estimating chimpanzee demography, social structure, and territory range with camera trap.
Thanks for the introduction. So this is part of my PhD project. And I just wanted to mention that I'm working closely with the World Chimpanzee Foundation, which is an NGO that focuses on the conservation of West African chimpanzee. And I might refer to them later in the talk as the WCF. Monitoring is a process of measuring a state variable at different points in time, and usually for like conservation studies, it's more about abundance and density estimation. But for elusive species with slow life history, such as non-human great apes, it's maybe a bit tricky to estimate population changes. Large population at large scale and for a long period of time, population changes was able to be estimated for West African chimpanzee, but mostly because the change was drastic. It was an 80% decline in the population. And so if we wanted to look at short-term variation, it's more tricky. The analysis doesn't have enough power. So instead of focusing on abundance or density, we might want to focus more on like demographic data. And this requires individual recognition, and it cannot be done at smaller scale for non-human great apes. So there's different monitoring method to recognize chimpanzees. So first there's arbitration, but it's not something really like we can do for a conservation project. There could be genetic sampling, so you can extract the DNA from fecal sample, but they're hard to come by, and you don't have information on egg structure. So here comes the camera traps, and we can identify the individual through their facial features. So for those unfamiliar with chimpanzee sociality, I'm just going to give a few key features on chimpanzees. So they live in social groups with several males and female. They're highly territorial. They range in efficient fusion dynamic. And the phyllopatric system. So the female, when they reach sexual maturity, they will leave the group they're born in to immigrate in another group where they will stay from the rest of their life, while the male will remain in the group they're born in. And so reproduction events happen within the groups. And so, as I said, they have a slow life history. So the questions we want to answer are, can we identify different social group with a multi-group area thanks to camera trap? Can we uncover some of the features of populations, social networks, structure, from the social networks we draw from the camera traps? And how do they use that territory in our study area? So I worked in the Moyen-Bafing National Park in Guinea, and we had 2 study sites that we sampled in the same fashion. So the first site called Bakun, it's a 10 by 10 kilometer grid where we set 100 camera traps, so 1 in each kilometer square cell. The camera were placed opportunistically to maximize the capture of chimpanzees, and we were recording videos. So we sampled from February 2018 to January 2019, and on the second site, Kutamba, we sampled so in the same way from March 2019 to March 2020. And the environment there is gallery forest along the rivers, grassy savanna and plateau, and in between it's bushy savanna and clear forest. So we're more on the drier type of environment where chimpanzee lives. So we recognize the chimpanzee, and to see if we were able actually to recognize them, we did some inter-observer reliability tests. So first, between analysts from the WCF, we set like a design with 32 individuals to match, and they independently say if they were a match or not. And we also did a comparison with the Chimpanzee Project. So it's a citizen scientist platform that was set up by the PANAF program. And so a group of citizen scientists also match the chimpanzees. We did 2 with the World Chimpanzee Foundation, and we compared like 50 videos, a subset of videos. And so we computed the Cohen's-Kappa coefficient, and we had a perfect agreement for 2 analysts and substantial agreement for 2 other, and we had substantial agreement for between the WCF and the chimpanzees. So we are quite confident in our ability to recognize the chimpanzees. So now we recognize them. We wanted to identify the different social groups. So we identified 227 individuals in Bakun. We built an adjacency matrix, so it's just an individual in the column and in the line. And inside the matrix is just the number of times each individual has been seen together. We use the package iGraph in R to just build the social network. And this is what we got. So in black is the position of the camera traps, the color dot are the individuals, the lines are with whom they were associated. And so there are 6 different groups that we identify. So 4 main groups in the middle and 2 peripheral groups. And we did the same for Kukutamba, and then we identify 5 different groups. So now the question is, we identify the different groups, but how do we know if we captured all chimpanzees in the group? So it's a tricky question. We don't really know if we managed to identify all the chimpanzees, but we can have an idea how close we are by drawing some accumulation curve. So it's just the number of days since the beginning of the study and how many chimpanzees we capture at each point of the study. And so if the curves reach a kind of asymptote, we can be quite confident that we're getting close to having identified all the individuals. This is what we get for backwind. So we kind of confident that it was a near complete identification. For Kukutamba, we had 2 groups where we had the same asymptotic shape, but for 3 other groups, it was more a linear increase. So we just didn't consider those one. So we made just a demographic table for the 6th group, and basically the medium number of the total number of individuals in each group, it's following the medium number comparing to other chimpanzee groups. The sex ratio was skewed towards male, which is quite rare for chimpanzee, but it's not uncommon for chimpanzee living in dry environment. And then we had quite a high ratio of non-weaned individuals compared to the number of adult female. And so to answer our first question, so yes, we were able to identify the different social groups. And thanks to a year of study period with the high density camera, we had a good knowledge on the different groups, on some of the groups in the study area. And if we were to repeat this design across years, we could access demographic parameters. Now about if we can get some information about the age and sex structure associations thanks to the social network, we draw with camera trap. So we applied the combined social relations and stochastic block model using the new strand R package. We specified the model for binomial data. We adjusted for the exposure, and we use a combination of age and sex as block. So, So basically, we obtained this. So we use the adult male and adult male association, as our reference point, because it's a phylopatric system. So we expect that we will observe more association between adult males because they're more likely to form bonds between each other as they stay in the same group their whole life. And so while we computed the contrast and the highest posterior density interval that we see, and if the contrast and the highest posterior density interval is lower and doesn't cross 0, it means that the probability of observing those associations in this category is reliably lower than the probability of observing association between adult males. And globally, we have mixed effect of age and sex structure on the association. But the association between adult males and female was less likely to occur than the association between adult males and females was less likely to occur than observing the association between adult males. And the association between adult female was less likely to occur or equally likely to occur than observing the association between adult males. So we're able to build a social network with co-occurrence of individuals, and it seems to support the theory that individuals of the non-dispersing sex form stronger bond. If we look at the territory, so this is what we can do with the camera traps is we can try to estimate just a minimum territory size. We just draw a minimum convex polygon around the cameras that captured each group. So obviously we restrain in our study area and where we place the cameras, but globally we had around 15 kilometers square minimum size. If we compare to another habituated group in the savannah, it's the same number of individuals, but they have way bigger territories. And we also computed the densities through capture recapture. And so they have a very high density in this region compared to what we would expect. And so probably the environment hold more food than expected. And so to conclude, camera traps can be a great tool, an alternative to monitor several habituated groups with a cost and time efficiency. With a 1 year long study period, we're able to near complete description of several groups. we could apply like capture-recapture type of modeling. And so yeah, we sampled, so we keep sampling in backroom. So now we're working on rematching the chimpanzee and try to estimate some demographic parameters, see which individuals survive or not. So thank you to my collaborators and yeah, everyone for your attention.
We have time for a couple of questions.
Hi, thank you for your talk. So I was thinking about what you said now at the end of across years identification,
yeah,
and thinking about the young and when they. So can you estimate any kind of young survival when they grow up? I guess it's harder to know which ones survive the north, and so.
Yeah, so this is what, yeah, it's recognizing the juveniles, it's a bit hard, but as they stay with their mother, yeah, so it's what we're trying to do. The only thing we won't be able to see if it's an individual, whether a baby was born that we didn't see and he died before we could see it. But for like, yeah, we have quite a good match across the years now, and we recognize the juvenile. So yeah, that's the idea behind it. I don't know if I answered.
Okay, well, thank you so much for your time.
Thank you.
And our final speaker for the session is Liam Bailey, monitoring changes in carrying capacity through time to assess conservation outcomes in a large social crime.
Cool. Okay. Thanks everyone for being here. Before I start, I want to thank my co-authors, Oliver Hohner and Alex Cotiol, who were invaluable in this work. So I'm just going to jump straight into it. So as I guess we all know, population abundance is a really important variable that underlies a lot of metrics that we use to assess populations that are of conservation concern. So things like the IUCN Red List categories or WWF Living Planet report, they all rely on monitoring population abundance and trends in population abundance. And often when we see a change in population abundance, we would consider that to be some kind of an indicator of changes in the environment. So we might imagine a scenario where environmental conditions have increased, the solid line here, and that we see some increase in population abundance in the dashed line. And in this case, seeing population growth would be some indicator of concurrent environmental change. But of course, for a lot of species, the rate at which they respond to changing environmental conditions will not be this rapid. So you could imagine a scenario where environmental conditions have improved sometime in the past before we started monitoring, and they've since stabilized. And so we still observe growth in the population, but it's not an indication that the environment is improving during this period of time. And in fact, as long as the population is below the limits of the environment, we could even imagine a scenario where population grows, even though during that period, the environment is deteriorating. And so to kind of sum that up, we could say that observing trends in population abundance often cannot allow us to distinguish effects of particular population dynamics from those of current environmental change. And a bit of a problem here is that often conservation initiatives are actually trying to affect the environment. So supplemental feeding, culling invasive species, all of these are affecting the environment, but we are not actually monitoring it directly. And so I've been thinking a little bit about other metrics that we can think about in addition to population abundance that can help us track environmental conditions a little bit more directly. And today I'm going to talk about an old idea, carrying capacity. If we're able to estimate carrying capacity, then this should be independent of the population dynamics at the population that we're monitoring. And it should incorporate all of the environmental conditions that are impacting our focal species or population, and also account for potential complex interactions that might occur between environmental variables. And we often think about measuring carrying capacity as something that's fixed within the system, at least for convenience. But of course, it is also possible that we could try and measure carrying capacity as something that varies over time. This is something that's been done in the past. And in this case, we'd have what we're calling time varying carrying capacity. And we could use that as some type of proxy of changing environmental conditions. So to think a bit more or try this idea out, I've been working on data from the hyena project in Ngorongoro Crater. It's a population of spotted hyenas in this wildlife reserve in the north of Tanzania. And we've been collecting data there since 1996, and the project is ongoing. The type of data that we collect is really diverse. We have people in the field who are observing the behavior of the individuals. This allows us to understand a bit about their social structure, about mate choice in females, about male dispersal between clans. We also estimate demography, so we have some estimates of apparent survival, reproduction, and litter sizes. And we also get genetic information from our individuals, and so that means we have a reasonably complete pedigree of our population. And I actually think this is quite an interesting population to study because it's undergone quite a lot of change over the past half a century or so. So in the 1960s, Hans Kruk was one of the first people to report on this population, and he estimated that there were about 400 adult individuals in the population at the time. And when we started monitoring in 1996, the number of adults, which is the darker shaded area on this plot, had dropped to about a third of those that had been seen in the 60s. So some decline happened before we were monitoring. Since then, the population has increased and it's now reached back to similar levels that we saw in the 60s. But as I kind of pointed out at the beginning, so we've observed these changes in population abundance, but this doesn't necessarily tell us how the environment has been changing through this time. And so I was interested to see whether we could look at this question. How has the environment changed or not during the time that we've seen this increase in population abundance? And so one way we've tried to do this is by using individual-based models. So we use all of the observational information that we have from our study from 96 until now. We fit a whole lot of statistical models that allow us to estimate vital rates and also male dispersal in between the clans in our study system. And in our vital rate models, we try and account for density dependence, for senescence, for social rank, which is important in hyenas. And we also try and account for environmental stochasticity by including a random effective year in our vital rate models. And then we also integrate all of the information we have about the kind of behavior of this system. So female mate choice, male dispersal, and the social hierarchy. And when we combine all of these things together, we get what we've been calling our spotted hyena individual-based model, or SHIM, which we hope can be used for actually a lot of things. So the key thing here is that because we've included our year term in the model, what we try to do is to do some simulated experiments of what the population might look like in different environmental conditions. So we could run our individual-based model where we fix the conditions to match those that are in a particular year. Let's say the year 2000. And yes, the GIF worked. And then if we start with a reasonable population size at the beginning, it should increase and eventually stabilize. And this point at which it stabilizes, we would take as some proxy for the carrying capacity at that year, in that year. Of course, we don't do this once. We can do it many times. And so in this example, in the year 2000, we would expect that the crater could have supported around 730 hyenas. It's important to point out here that we haven't imposed carrying capacity at all here. This is all just based on the emergent properties that come out of the model because of the density dependent terms that we estimate in all of our vital rate models. So if we go back to our change in population abundance, so this is the blue line here is the observed change in population abundance over time. And then the red line here is our variable carrying capacity that we estimate in all of our different years. And what we see is that, maybe unsurprisingly, it's varied quite a lot, but we don't really see any clear temporal trend in carrying capacity. And in fact, the median value of carrying capacity that we estimate over all this time is quite similar to what we get if we just assume that it remained totally stable the whole period. And so this then would suggest that the changes we've observed, the growth in the population since 1996, is not an indication of current changes in the environment, but something that's happened in the past, some improvement that's happened in the past. And then we kind of thought at this point, well, if we're seeing this variation in carrying capacity, are there any environmental variables or variables that we've measured over time that could help us understand why or what drives this change? And we did some simple analysis of this. This is pretty preliminary, but it seems that prey abundance and prevalence of disease are the key factors that lead to variation in carrying capacity in the system. And this fits exactly what we would expect from what we know about the biology. But it's also important because both of these variables are things that we would expect to change as climate change affects the East African savanna ecosystems. So to conclude, measuring population abundance is often practical because it can integrate a lot of things, but it has limitations, especially in cases where the thing that we're interested in is actually the environmental conditions themselves. If we have sufficient demographic data, we can try and more directly estimate environmental change, perhaps using methods like I've presented here today. And this hopefully allows us to better understand our focal populations, and also I think can help us better understand the outcomes of conservation interventions. So we can see if when we do something that we hope will improve environmental conditions, whether environmental conditions actually change. In our hyena system, we saw a lot of variation in carrying capacity, but no clear trend. And as I said before, this seems to be driven by disease and prey abundance. And so these are now topics that we want to focus on a bit more so that we can understand things, the variables that we think will be really important to try and maintain this population into the future. Thank you very much for listening.
We have a bit of time for questions.
Thanks, Liam. Really cool talk. I was amazed that the precision you were showing on the carrying capacity estimates was like remarkably high.
Yeah.
And so I was wondering does that only reflect the stochasticity in the IBM, or do you have a way of propagating parameter uncertainty or even kind of calibrating the IBM to data?
So, yeah, I think that, I guess for full transparency, when we first started this estimation of carrying capacity wasn't our original goal. And so I think that the underlying models that we use are still fairly simple. And that means that there may be more uncertainty in our vital rates estimates, which would lead to further uncertainty in our carrying capacity estimates. We don't yet have any, we haven't kind of looked into that step yet, but I think it would be important to, if we need to use this more. Having said that, the model really behaves, kind of the data that we see in the model behaves quite well with what we would expect. You know, we don't get scenarios where you get a population growing when it's above carrying capacity. We also do population pattern-oriented modeling with our IBM to make sure that kind of all the patterns in lifetime reproductive success and survival all seem reasonable. So I think it's okay, but I think you're right that there's probably uncertainty that we're not accounting for yet, and we need to think about that.
Okay. Well, thank you so much, Liam, for your talk. And with that, let's thank all of the speakers from the session.
But anyways, the thing that we really so is this assumed matrix strong. It's been hard to yes, yes, and our like preliminary studies that we looked at mentioned and about 70% and then you get this unity where it's like maybe males are leaving, so although we know we would here is a like choice, but it's actually male choice that we're and yeah, you could totally
Elle est connectée?
Non, mais ça va, alors Alpha, il y a 1 minute 30 de... Mais il y a pas, et en vrai, il y a pas mal de gens qui ont fait 9 minutes et...
Ça c'est des questions là. D'accord. Il faut jouer de la discussion ici, là. Ce qui est autre chose, en fait.
Allô, bonjour.
Bonjour. Ça va, et toi, Olivier? Olivier.
Voilà, ça aurait bien.
Oups, décalé. C'est parti.
Hello, everyone. Hi. Thank you so much. So we're going to start the last session of talks, which is entitled Management and Conservation Part 2. And we're going to start with Kylie Dunham, who is affiliated at the Eastern Ecological Science Center at the US Geological Survey, and she will talk to us about design and evaluation of long-term monitoring programs to inform annual life cycle modeling, conservation, and adaptive harvest management of migratory waterfowl in North America.
So I'm going to talk about some of the work that I've done designing and evaluating monitoring programs for individually marked migratory waterfowl in North America. Waterfowl banding and dead recovery data led to the development of the flyway system that's been used for managing migratory birds since 1950. Ongoing banding efforts provide the information that's required to conserve waterfowl habitat and estimate parameters used in the adaptive harvest management program for setting duck hunting regulations. The banding program is coordinated annually by the U.S. Fish and Wildlife Service with help from a number of other agencies. And waterfowl are primarily banded during the pre-season. This is about August to September, which occurs after breeding and just prior to harvest. Banding sites have varied over time, but many are in Canada and across the northern United States, and thousands of ducks are banded annually at each site. The banding program has been largely successful. However, there are several limitations and concerns that triggered this project. Namely, the data collected per marked bird is low across space and time. We rely on dead recovery data only and do not include recaptures. And harvest and the probability a hunter reports a recovery have changed over space and time. The waterfowl management community would like to switch from separate harvest and habitat management frameworks towards an integrated management plan. This would require considerably more information on the birds throughout the full annual cycle, and the current program does not provide the required data. Additionally, there have been many advances in technology and modeling that have improved our inferential abilities, and these have yet to be applied to this problem. And so the Fish and Wildlife Service wanted to know if there was a better way to monitor individually marked waterfowl to inform conservation and management. To address this question, we use a structured decision-making approach with participants from the Migratory Bird Management Program within the United States Fish and Wildlife Service. Structured decision-making is used to provide insight and guidance to decision makers and generally follows the steps also known as PROACT. These steps include specifying the context and defining the problem that we need to address, which we've done here. We identify the objectives and assign measurable attributes, then develop alternatives or look at the different options we're going to consider, then quantify the consequences of each alternative and look at how they perform in respect to the objectives, and finally evaluate trade-offs and select the best actions. Participants identified two objectives for this case. First, they wanted to minimize long-term costs, so we estimated the expected cost of different marker types, the cost of data retrieval, and any costs associated with additional required equipment. Second, they wanted to maximize the amount of information that was gained from marked individuals. To quantify this, we determined the number of estimable parameters and multiplied that by individual parameter weights. And these parameters were weighted by importance as ranked by the participants. So to do this, I elicited parameter weights to reflect the relative importance for adaptive harvest management, integrated management, and other parameters of interest. 4 parameters had to be estimable under all alternatives in order to inform the current adaptive harvest management program. And these were assigned a weight of 1. The additional parameters were assigned lower weights based on their contribution to information needs for an integrated habitat and harvest management approach or for other parameters of interest. To develop alternatives, I conducted a literature review of available technologies that could be used for monitoring individually marked ducks. We also considered study design and analysis elements. Then we constructed a simple strategy table like this that listed out the available options, and we created alternatives based on combinations of marking technique, capture season, and counter methods in the data type or analysis we expected to do. We ended with a lot of alternatives and pared it down quite considerably. We have the alternatives listed here on the left, and on the right we have our objectives. Again, we wanted to maximize the amount of information, which we measured as a function of the number of estimable parameters and their importance. And we also wanted to minimize costs, which we estimated over 25 years. Here, the purple cells performed the worst within their category, green performed the best, and white was about a middle ground. Importantly, the status quo, which is our current banding program, is amongst the cheapest, but performed the worst in terms of the amount of information that was returned. And the main takeaway from this portion of the assessment is that there was no clear winner, and this brought us to our trade-offs. So I'm going to highlight 2 alternatives that participants were particularly interested in and discuss the trade-offs between the 2. Alternative 4 included shifting from the single pre-season banding towards a formal 2-season banding effort and included recording recaptures at all banding sites. So now we have dead recoveries and physical recaptures annually. Under this alternative, we determined that we could estimate more parameters than with the status quo, but unfortunately are not able to get at some of the parameters that are required to implement an integrated management approach. And then information on migration and breeding was generally lacking. This alternative, however, is relatively cheap, but did not improve the number of estimable parameters by much and prevents an integrated management program. And so we needed to consider if the small increase in information is actually worth those additional costs. For alternative 14, this would be sticking with a single season banding, so that's our pre-season marking. However, we're adding MODIS tags on a large subset of individuals. We're also recording physical recaptures at those banding sites, and this would also require us to establish a network of MODIS towers in high priority areas across North America and develop an integrated model that accommodated all of these data types. Under this alternative, we determined that we could estimate more parameters than with the status quo, including those that are required for integrated management in addition to several other parameters of interest. However, this alternative is substantially more expensive and logistically very challenging. So in this case, we need to determine if it's more important to increase the information we gain from the monitoring program or if it's more important to keep costs down. So the major trade-offs the service had to consider were between cost and the amount of information provided, but several other things came up while reviewing alternative performance. Integrated management is a priority for the service, but to get the required information is going to cost a considerable amount of money. So we need to consider if it's actually worth the financial investment and is it really necessary for more effective management and conservation of the species. Further, for the MODIS alternatives, we identified several interesting trade-offs or considerations, including the logistical challenges of establishing a network. We also identified several opportunities for cost sharing and multi-taxa conservation. So realistically, the service would work with partners to establish a large-scale monitoring network that could help monitor many species across North America. So using this framework, we were able to streamline the decision-making process and formally consider alternatives to a long-term continental scale monitoring program. Part of the motivation for this project came from the huge number of options available for monitoring individuals with new technologies, but the service couldn't make a decision about changes to this program without understanding how these options compare to one another or how they performed against their objectives. Working through this process, a lot of the alternatives participants expected to rank really high were effectively eliminated as infeasible, and others they expected not to work performed relatively well. So overall, this process really helps the service to assess the potential for changing a critically important monitoring program in a transparent and defensible manner. All right, so with that, I will gladly take any questions, and I also included my contact information down here on the left, so we can go with my personal email, my Cornell email, or you can tweet me at Kylie Dunham on Twitter.
So before we start for the questions, Kylie is online, but she sadly has no voice anymore, so she cannot talk. But she offered to have answer one question and then write the answer through the chat. So we might need a little bit of time for her to write her answer. So yeah.
Yeah, I was wondering if crippling glass was among the things investigated, because it's a major component of hunted of quarry species management.
So first, Kylie, could you hear the question? Can you answer on the chat if you heard the question? Make a sign. Okay, yes, she answered. Yes, I heard. Okay, so I guess she's typing. Someone has some elevator music in the meantime. I'm good, thank you. How are you guys? Good? Enjoying the conference so far? Okay, she answered. Okay. She says, we discussed crippling loss, but decided against including it as a parameter of interest. However, in the future, we expect we will review that parameter because it is rather critical, exclamation point. So I guess in terms of timing, we're good. We could go for a second question, I guess. Oh. Okay, so I guess we will go to the next presenter. So next is Chris Sutherland, who's from the University of St. Andrews. And he's going to talk to us about the opposite effects of global and local density-dependent dispersal on metapopulation stability and persistence.
Hello, today I'm going to talk about density-dependent dispersal in spatially structured populations and the consequences for metapopulation dynamics and persistence. And I'm going to do this using a classic metapopulation of waterfalls in the northwest of Scotland, where we've been working for the last 20 years, monitoring 110 discrete habitat patches using latrine surveys to give us information about patch occupancy and live trapping to give us information about local abundance. And this is what the data look like from this 20-year monitoring effort. On the left-hand side, we have the metapopulation dynamics. That's the proportion of the patches that are occupied in each year over time. And on the right-hand side, you see the spatiotemporal dynamic local turnover rates. So we have all the green that are colonization events, and all the red dots you see are extinction events. And the reason we refer to this as a classic metapopulation is because of this long-term fluctuation around an average metapopulation size, and also this high degree of local turnover, lots of colonization events and lots of extinction events. So you have metapopulation dynamics being driven by colonization extinction dynamics. So if you have classic metapopulation data, you turn typically to classic metapopulation models. And the model I'm going to be using today is what I refer to as a hierarchical stochastic patch occupancy model. So this is hierarchical because we acknowledge that we don't see the patch occupancy states perfectly. So we treat those as latent or partially observed states. And because we have repeated visits, especially in more recent years, we're able to develop an observation model that links our observations, our imperfect observations of detection, non-detection data to the true underlying state. The state model is Markovian, where the probability that a patch is occupied in any one year is conditional on its occupancy state in the previous year. So for example, a patch that was occupied in the previous year is predicted to be occupied in the following year with a probability of 1 minus extinction, which is the survival probability. And an empty patch last year will be occupied with a probability equal to the colonization probability. Now, the nice thing about stochastic patch occupancy models, and the reason they are the analytical core of spatially realistic metapopulation theory, is the way that we formulate these or parameterize these extinction and colonization rates, or these turnover rates or turnover probabilities. First, we link extinction probability to population size. So this is the idea of demographic stochasticity, where we assume smaller patches are more likely to go extinct than larger patches. Then we have a function that maps colonization probability to connectivity. And really, it's connectivity that is the workhorse, or the spatially explicit workhorse of the stochastic patch occupancy model. It's what gives it its spatially realistic characteristics. So here I'm going to focus on this connectivity term S. So the connectivity of a focal patch is essentially the sum of all the contributions from all the neighboring patches in the network. And we can characterize these contributions using a kernel, a dispersal kernel. So here we have a half normal dispersal kernel with two parameters, rho naught, which we interpret as the effective dispersal rate. And we have this parameter sigma here, which determines the spatial scale, and which we interpret as the typical dispersal distance. So now, once you estimate these parameters, what you're able to do is quantify or characterize the contributions of neighboring patches to a focal patch's connectivity based on the distance, the pairwise distance. But in addition to the spatial weighting, we also weight these contributions by the occupancy state, just making sure that only occupied patches can contribute to connectivity, and then also by local abundance. Again, here we're assuming that larger patches are going to contribute more to connectivity than smaller patches. And because this is the spatially explicit core of the stochastic patch occupancy model, this is where I'm going to focus my efforts for the rest of the talk, and where I'm going to start introducing the idea of density dependence. Because the standard parameterization of this connectivity term has this fixed rho naught parameter here. So the typical standard way we model this is to assume that this parameter is fixed, constant in space, constant in time. So what I want to do is relax this a little bit and model this parameter as a function of covariates, and those covariates are going to be specifically selected and related to density at a couple of spatial scales. So here we're going to extend this model out using a log linear model for this dispersal rate. So the first covariate I'm going to introduce here is local density. So this is a term that's going to capture our local density dependence. And D here is the population size in the focal patch or in the source patch. So here we're relating dispersal rate to the dispersal rate from a patch to the density in that source patch. And here beta 1, the estimate of beta 1 is going to be the strength and direction of the local density dependent effect. In addition to the local density dependence, we're going to add a second covariate, which is I refer to as global density dependence or landscape level density dependence. So MT here is the metapopulation size, or the proportion of patches that are occupied in the previous year. This is a proxy for available breeding territories on the landscape. So this is the second scale of density dependence, and we have beta 2 here, which is the strength and direction of global density dependence. So fitting this model to the waterfall data, what we find is strong empirical support for positive local density dependent dispersal. That is, the probability that you leave a patch and disperse from a patch increases as the density in that patch increases. So this is classic positive local density dependent dispersal or density dependent emigration. In contrast, we find support for negative global density dependence. That is, as the metapopulation fills up, or you can think of it as the number of available territories decreases, that dispersal rate decreases. And this window here just represents the range of values that we actually see in the data. So what does this mean for metapopulation dynamics? So we have a model where we can model density dependent effects on dispersal, but what difference does that make to, or what are the implications for metapopulation dynamics and persistence? Well, in order to explore this, I fit the model and then projected the model 20 years into the future and monitored the behavior. In particular, I was interested in the metapopulation growth rate. And what we find is that by having density dependence at these two characteristic scales, what emerges from that is strong negative density dependent metapopulation growth. So here you can see one projection here, and that projection fluctuates around the long-term average due to the strong forcing back to the average, this stable point. And this strong negative density dependence means that we actually observe very few metapopulation extinctions over this 20-year period from these thousands of projections. Now, although I find this interesting in and of itself, what is instructive to do is compare this with another scenario, namely a scenario where we have only local density dependence. Now, the reason this is interesting is that local density dependent dispersal, so when we have dispersal that's a function of the density in the patch from which you're dispersing, is widely documented to occur in natural systems. And we also know that it has a destabilizing effect. So here what I've done is I fit the model with both density dependent terms and then another model where we have local density dependence only. And what we can see is already you see that larger variance in these projections relating to the destabilizing effect of local density dependence. So what about if I compare the growth rates? Well, what we find, as is demonstrated by this trajectory here that doesn't seem to be attracted to this long-term average, we essentially have density-independent metapopulation growth relative to the strong negative density dependence that you get when you have the both scales. So what we see here is that the dual scale density dependence is actually stabilizing, despite there being the presence of destabilizing local density dependence. So what does that mean for metapopulation extinction? Well, actually, when you have local density dependence acting on its own, you get large, up to 25% probability that the metapopulation goes extinct. And that's in contrast to the vastly reduced or vastly buffered situation where you have both scales of density dependence operating. So what we see here is that having these two scales of density dependence actually buffers against the extinction that is expected when there's only local density dependence occurring. So what I've done is use this hierarchical SPOM stochastic patch occupancy model to investigate dual scale density dependent effects on dispersal. We're able to find empirical support for these scale-specific effects on dispersal, namely positive local density-dependent dispersal and negative global density-dependent dispersal. And by adding those density-dependent terms on dispersal, what we end up with is emergent metapopulation dynamic processes, namely strong negative density-dependent metapopulation growth. And this actually acts, by having the second scale, acts to offset the known destabilizing effects of local density dependence. Essentially, what we think we find here is a mechanism for metapopulation persistence despite the presence of destabilizing local density dependent dispersal. Okay, thanks very much.
I can't hear anything. Okay, you can hear me, but I can't hear you. Maybe we can do the reverse of Kylie and someone can type a question if there's time. Actually, Charles, I did use log. I used log of the population size for local density dependence. I just size for the local density dependence. That's why there's the effectively what looks like nonlinearity in that surface that I showed. During the workshop, Todd. Thanks, everyone.
Je pense que c'est la vidéo qui a bugué. Ouais,
Everybody? The sound is working again, so we are back to the last two talks. Are we ready to get back to it now? Just a little moment. We have the sound. All right. So now we will welcome Eva Conquet. Is it remote as well or in person?
No, I'm here.
So sorry. So Eva is from the Department of Evolutionary Biology and Environmental Studies at the University of Zurich. And she will talk to us about the effects of season density interactions on the vital rates of an African lion population.
Yes. So is it the PowerPoint or is it the PDF? Can we put, because I think I sent, sorry. Good. So I'm happy that Julie here doesn't have to type my whole presentation on Zoom. I'm going to talk to you about some results from one of my PhD chapters on the effect of season density interactions on the vital rates of the lions. So we know that vital rate variation can arise from very complex mechanisms. And usually in population ecology, we look at the effect of the environmental variables on vital rates. But the environment can be autocorrelated. And sorry, I don't know what is appearing weirdly here, but I hope we can still see. So this environment can be spatially and temporally autocorrelated, and vital rates can respond non-linearly to these environmental variables. So vital rates affect population dynamics, and these population dynamics can in turn create feedback loops through density feedbacks, either directly onto vital rates or through interactions with the environment, mediating the action of the environment on vital rates. So for my talk, I will focus on density feedbacks and their interaction with the environment, and for the autocorrelation part, we'll have temporal autocorrelation with seasonality. No worries. So usually density dependence and so the effect of density on vital rates is assessed using population size or population density or group size or density, or the two of them together. But there are many other density measures that we can use to assess density dependence of vital rates. For example, how another group affects a focal group, how a subgroup within a group affects another subgroup, or how a subgroup affects another group or another subgroup in another group. And so the question here is whether accounting for these socially and spatially explicit density measures can help us better understand how density dependence, how density affects vital rates and in turn population dynamics. So to answer this question, I will show you some results on a population of African lions, and more specifically, a population that lives in the Serengeti National Park in Tanzania. And the main characteristic of the environment of these lions is that it is seasonal. So we have a dry season and a wet season. So we saw before the chimpanzees' social system, and the lions work a bit the same way. So we have lion prides with a resident coalition of males, the females that stay in their nature pride most of the time, and their offspring. And then we have coalitions of nomadic males that roam around and try to find a pride from which they will be able to take over, kicking out the resident coalition and be able to reproduce with the females of that pride. So when the nomadic males manage to kick out the resident coalition, they will become resident, the evicted coalition will become nomadic, and the new resident coalition will kill the young individuals to be able to reproduce faster with the females. And so here I assess the effect of density using density measures at 2 levels. The socially explicit level with at the group level with the pride and resident or nomadic coalitions, and the spatially explicit level with the home range level. And I assess the effect of these density measures for each of the dry and the wet season. So this is the life cycle that we use to estimate to build the model and estimate the parameters. And so we have the young subadults that become old subadults, either male or female. And the female, when they grow, they will become female and reproduce. And the males will become young males that stay in their nature pride. And once they emigrate from their nature pride before they are 4 years old, they will become either resident or nomadic. And the resident and nomadic adult male switch states according to the eviction and takeover dynamics that I showed you before. So to estimate the survival and transition rates, which correspond to these colored arrows here, we used a Bayesian multi-state capture-recapture model that was based on what Chloe did during her PhD on the trouts. And we used capture histories on more than 1,300 lions across 30 years. And for the socially explicit level, we used the number of females in the pride and the coalition size of a resident or nomadic coalition. And for the spatially explicit level, we used the number of nomadic coalitions in the home range of a pride. And as I said before, we assessed the effect of all these covariates for both the dry and the wet season. For the reproductive rates, which correspond to reproduction probability and recruitment to 1 year old, we used a Bayesian generalized linear mixed model. And here we used data on about 500 lionesses collected over 30 years, and the same covariates still at the season-specific level. So let's have a look at some results. One of the main results here is that adult female survival appears to be density independent. And this could be a very important thing because in long-lived species, adult female survival is often one of the vital rates to which the population growth rate lambda is the most sensitive to. And so if there is especially no negative density dependence on the survival of adult females, it could be a way to buffer the negative effects of density dependence. So for the effects of socially explicit density, you see that the survival of young subadults decreased with the number of females in the pride. And this is because in big prides, there are a lot of females, so lots of opportunities to reproduce. And so the nomadic coalitions will focus on these prides to try and take over. And when they manage to take over, they will kill the subadults. And for the nomadic males, their probability to take over a resident a pride increases with the nomadic male coalition size in the wet season, but doesn't depend on the size of the coalition in the dry season. And so in the plains especially, so in the plain habitat, the takeover season is mainly in the wet season. And so in that season, if you have a bigger nomadic coalition, you are more likely to be able to take over the pride because you are more likely to be bigger than the resident coalition that you are challenging. For the spatially explicit effect of density, we find that the survival probability of young subadults decreases with the number of nomadic collisions in the home range in the wet season, but not in the dry season. And again, here, so the wet season is the main takeover season, and the more nomadic collisions you have in the home range of the pride, the more likely they are to take over the pride and therefore to kill the subadults. And similarly, we see that the eviction probability of resident males increases with the number of nomadic collisions in the home range in the wet season. So I didn't show you all the results, obviously, just a subset of it, but this is a summary of all the results that we find. And so you can see that there are very complex effects of the socially and spatially explicit density measures that affect different vector rates in different ways. And there is also sometimes a great difference between how these density measures affect vector rates depending on the season. So the next steps will be to fill in the gaps on the missing parameters that we were not able to estimate with these models using data such as previously published data or expert knowledge. And then project the population using a spatially explicit individual-based model and use that model to assess the importance of density feedbacks in the lion population. And with that, I would like to thank you all for your attention, the whole organizing committee for this wonderful conference, and my supervisors, my collaborators, especially Chloe, who practically saved my PhD with her help with the models, my committee members, especially Olivier with his wonderful feedback every time we meet every year, and the institutions that fund and support my PhD. And I know that this is the last day, so this is how you might look like now. So don't worry if you don't have any questions, but I'll be happy to take any if you have some.
Thank you, Eva, so much for this interesting talk.
Thank you.
Are there any questions?
So I'm just kind of interested in what you think after we saw Marlene's talk plenary previously about also the competition with like density dependent effects of heterospecifics, because in the Serengeti you could also get information about the hyena numbers. That's obviously a lot more complex, but I'm just kind of wonder how you think that would play into the dynamics that you're already seeing here.
Yeah, so I think one of the main things, so I think the lions sort of outcompete the hyenas most of the time. And I think in terms of interspecific interactions, what would have the main effect would be the prey availability. So the wildebeest, especially because you have this whole migration. Unfortunately, I wasn't able to get any like usable data to put in the model. So yeah, but it would have been very interesting to look at.
We have time for one more question.
Do you have hunting in this population? And if so, do you know how it could affect the fission dynamics?
Yeah, so I think there is, or at least there used to be trophy hunting. And there have been previous works that have looked at what happens depending on the age of the male that you hunt. And they found that you would have to hunt for I think the younger males, or not the older males, because if you hunt for the young ones, then you disturb those dynamics and you increase the mortality of the young, etc. So, but yeah, we didn't include it here, but hopefully maybe in the individual-based model.
Okay, thank you very much.
Thank you.
And now to our next speaker, we have Nicolas, so sorry, pardon me if I pronounce it Strebel?
Stable, yeah.
Strebel, from the Swiss Ornithological Institute, who's going to talk to us about multi-species population indices for sets of species that include disappearing or newly occurring species.
Thank you. Right. Over time, it's just common that some species disappear and new species arrive in a study site. And with the current methods on calculating multi-species indicators, zeros are a bit hard to include. And so I will talk about methods that allow including a disappearing species or newly arriving species because they are also part of the aviform or of nature in general. With multi-species indices, I mean multi-species population indices, like, for example, Living Planet Index or diverse wild bird indicators. Typically, until now, the geometric mean was used to calculate these MSIs because the geometric mean gives equal weight to all species. And it results in trends in average relative population size rather than absolute population size. So based on geometric mean, we can, for example, say that today's populations are 30% higher compared to 1990. This example is about breeding birds in Switzerland. There are some disadvantages of the geometric mean. One is that rare species typically show much stronger fluctuations in relative terms. So the increase of 3 pairs here has a much stronger influence on the MSI than the increase of 20,000 in the abundant species. And even more problematic, the geometric mean actually collapses to 0 if at least one species in 1 year has a population of 0. There are different methods, or people try to overcome that differently. For example, you can, in your MSI, only include common species, but I think this is not a good idea because this leads to bias in terms of rarity in your species group. So that's not really representative. And you could also just ignore 0 values, but this leads to species selection changes, and that means that you can't really compare different values or values from different years if the species selection changes. Another thing is to replace zeros by small values or to generally add small values to your relative population indices to avoid zeros. But this leads to an arbitrary influence on the MSI. The figure here shows a curve of an MSI of about 20 farmland species in Switzerland. And among these 20, 5 species disappeared between 1990 and now. And as you can see, the shape of the curve looks very different when you replace the zeros by different values. And this curve gets some attention by the general public, also by politicians, because it's about farmland birds. And before seeing ourselves in the eye of the storm, I said, okay, we have to look for alternatives without or with less arbitrariness. And so actually we realized that the log normal model is a statistical model that produces geometric mean. The linear predictor mu it equals the year-specific intercept, beta t, and species can be included as factors. And then the exponential of the year-specific intercept, beta t, equals the geometric mean. So it's kind of a model-based approach of the geometric mean, but again, the log normal model cannot deal with zeros, but motivated by the finding, we thought that, okay, maybe we can find other models that can deal with zeros and that produce similar multi-species indicators. And so two alternatives that we looked at fitted these criteria. One was the negative binomial MSI, and the other was the compound Poisson. The compound Poisson is, or the correct term for the distribution for the compound Poisson is actually 3D compound Poisson. Call it compound Poisson from now. The compound Poisson is fitted to annual relative population sizes, like the geometric mean, and the negative binomial model is fitted to annual absolute population sizes. Typically, both are estimates. The 3D compound Poisson distribution is a mixture of a Poisson distribution and a gamma distribution, and this has mass at 0 and supports non-negative values, and is, for example, used to model means of counts. I'm sorry. The negative binomial distribution is suited for count data with variance differing from the one expected by the Poisson distribution, and it supports non-negative integers. In both models, we have year and species as predictors, and the multi-species indicator is derived from the year-specific parameters. Then we wanted to look at how sensitive these new methods are in different scenarios, how they differ from the geometric mean. And so we simulated some data. We simulated data for 20 species, 10 years. Part of the species group has a strong positive trend. Part of the species group has a moderate negative trend. So the number of species with a strong trend was varied from 1 species or 5 species to 10 species. And the initial population sizes, we also varied from 10 to 100 to 1,000 and to 100,000 for the species with strong trend, and the other we always set to 1,000. That's what the results look like. These are the species-specific population developments based on these simulations. And then we fitted MSIs. And so what you can see here is that for some scenarios, the multi-species indicators look quite similar. So in case there are very few species with a strongly diverging trend, then the result is quite similar. The curves are more or less the same. And also if the species with a stronger trend do not show too strong fluctuations, then the curves also look rather similar. In contrast, if we have some species with a very strong trend, but that show strong fluctuations, because for rare species, as we simulated it, the fluctuations are much stronger, then the negative binomial kind of filters them out or ignores them a little bit. Thank you. And Poisson compound and the geometric mean follow them more tightly. On the other hand, if the group of strongly increasing species shows less fluctuation, then the compound Poisson picks that up more, stronger than the geometric mean and the negative binomial. Then we also wanted to look at the influence of single species. That's a group of alpine breeding birds in Switzerland. It's the same figure. This is population size, log scale now, and trends over years for species. Geometric mean would basically be the mean value of these curves per year. And then for the comparison, we also looked at other groups. What I show here is just briefly in black, the multi-species indicator, and in red, the multi-species indicator if a species is dropped from the group of species. So for forest species, the curves generally look different for the different methods, and also the influence of dropping one species is not that big. Because these are mostly common species that don't show that extreme fluctuations, but also because the group is rather big, so 56 species. On the other hand, if you look at the wetland species, dropping single species has a much stronger influence if you use the geometric mean compared to the other MSIs. And for the alpine species, particularly when you drop the bearded vulture that started breeding in 2007 and thus shows a strong positive relative trend, then this species has an extremely strong influence on the geometric mean, a bit less so on the compound Poisson, even less on the binomial, negative binomial MSI. To summarize, the methods have advantages and disadvantages. The new methods can deal with zero, what is an advantage for us. They are less influenced by single species and also less influenced by strong fluctuations, what we think might be good. On the other hand, the geometric mean is well understood, generally well understood, and therefore also easier to communicate. To conclude, there is no silver bullet. If all species are common, you might use the geometric mean MSI, but probably evaluate the influence of single species on your MSI. If you do have some rare species, new or disappearing species in your set, and that will always be the case over long time series, population sizes are not available, then you can use the compound Poisson. If population sizes are available, you can use either compound Poisson or negative binomial. In addition, we recommend to report the proportion of species with positive trend, negative trend, and newly and disappearing species. All right, that's about it. Thanks to Francie, she conducted most of that project. And thank you for sticking with me for the last couple of minutes of this conference.
Thank you so much, Nicholas, for this very interesting talk. We have time because also this is the last talk, so maybe we can take the time to have a couple of questions if we're not too hungry or too tired.
So thank you for the talk. So if I understand right, you provide a solution to keep the geometric mean indices, but dealing with problems with small numbers, if I summarize. And so I was wondering, what are your thoughts on the geometric mean as an index? Like, it does two things at once. It measures abundance, and also it's meant to measure diversity. And we could also invest in looking at different sets of indices that measure different things separately, like diversity indices and population abundance. And why should we invest in using more the geometric mean? So my worry is we have now a better index that people will be able to use on even more data sets and use the geometric mean in even more context. Do you think there's a danger there, having this one index?
There's danger in having basically the geometric mean index instead of other diversity-based indices. I think generally, maybe not too simple to answer, but generally the geometric mean is not a bad thing because it is abundance-based and of course still kind of how we adapted it, it can also include aspects of diversity. Yeah, maybe that kind of answers your question, or we can talk about that a bit more in detail later. Yeah.
Did you have any examples of species that blinked in and blinked out? So like went extinct but came back in, or any insights about how those would be handled by any of the three models?
that didn't breed in some of the years, for example.
That disappeared for a couple of years, came back, disappeared again, so like both reappeared and went extinct.
Yeah, I think that's a similar problem with species that just disappear or appear in general, and always leads to a multi-species indicator of 0 in years where they are not here if you use the geometric mean. But the other methods can deal with that. Yeah.
Thanks for the talk. Have you had, or I assume you also have cases where their data may be missing for some species for like a year here or a year there. It's like how are the NAs handled in the indices you calculate?
Well, I didn't really talk in detail about the implementation of the method. Basically talked about the method, but when we implemented the method, we also thought about things like that. For example, indices always come with uncertainty, and either relative indices or also absolute annual population indices, and we try to deal with that via simulations. Not just fitting it once, but fitting it many times. And yeah, we also thought about method to how to deal with NAs in these indices. And yeah, apparently it works. Yeah, there are solutions.
All right. Yeah. Okay. Then thank you very much. I guess we can give a last round of applause for the people who presented this morning.
Okay, so okay, so we're going to go for lunch. But before we do that, just a quick word about the workshops this afternoon. So we have 3 workshops. One is going to happen here. Okay, the integrated model workshop, easy for you. And the 2 other workshops will take place in our building, in our research institute building. So the best thing to do is probably to split in 3 groups while we're having lunch, well, at the end of the lunch, okay? And we'll guide you to this building where 2 other workshops will take place, okay? So just follow us and you'll find the place with us. And because we all vanish after the workshop, we thought that it would be better to close officially the conference now. Okay, so I'm going to ask all the members of the local organizing committee to join here, to come down to the scene. And we're going to thank you very much for coming to Montpellier, for your contribution to the conference, for making I don't know, this whole week very enjoyable and friendly and everything. That was quite a ride, I think. I'm speaking for everybody, but yeah. Well, we're missing a few people, no? Okay. Do we have anything to add or no? Okay, so it's time for lunch. Yeah, where are you? Come down. Chloe, Sarah, Thomas, where are you, Thomas? And Maddie? Maddie, maybe? Ah, you. And Thomas is... He's left already? Okay. Okay, a round of applause for that. We're good? Okay. Thank you very much.